Welcome in our animal-microbe co-evolution lab at the Institute for Biodiversity and Ecosystem Dynamics at the University of Amsterdam. In our group, we study interactions between bacteria and the host organism they colonize, which is a small but iconic model organism, the nematode worm Caenorhabditis elegans, or C. elegans for short. These tiny worms are just about 1 millimeter long when fully grown. Because of their short generation time, of just 3 days from egg to adult, we can observe evolutionary change happening under our eyes. The nematodes feed on bacteria, which at the same time grow on and inside their body. In the wild, they live in bacteria-rich environments such as rotting fruits and plant material. Co-evolution of worms and bacteria is an important component of the soil ecosystem, which in turn is the basis of our food supply. The evolution of worms and bacteria happens at short timescales. Our environment is rapidly changing as the climate is warming and we're seeing more extreme weather events and using more pesticides. These rapid changes lead to the evolution of the worms and bacteria. And predicting the outcome of these evolutionary changes is important for ensuring global food supplies. Through our experiments, we learn how to make these predictions.
In our experiments, we try to better understand what makes some worms more successful than others. In evolutionary terms, we say that worms that are more successful have a higher fitness. The worm's fitness is determined by so-called life history traits. These capture, for example, how fast C. elegans grows, how fast it develops, and how many eggs it lays. In the lab, we create controlled microenvironments on petri dishes where the worms can grow on. We have to actually look through a microscope to see the worms because they are too small to see with the naked eye. Through the lenses of the microscope, we enter the odd microenvironment of the worms. Here, the worms go by their own business while we watch them and determine their traits to find out how these determine how successful they are in a certain environment. To create these different environments, we grow the worms in the presence of different bacteria, we increase temperature or salt concentrations. And then we measure egg numbers and their development over dozens of generations. Our goal in the end is to combine these measurements with information about how the worms change genetically. We then from that derive predictions about how they will evolve in response to environmental change based on these genetic information.
Identifying what we can and cannot predict in evolution is part of understanding the origins of the rich complexity of life on Earth. This is why our research is part of the Dutch Origin Center, a research initiative funded by the National Science Agenda. The Origin Center brings together scientists from across different disciplines to work together on challenging questions related to the origins of life. You can visit our website for more information and follow the Origin Center for updates on the exciting science that unites.